Alright, so what I want to do is follow up a little bit about our discussion in class today on growth. We're talking about this term growth, and it comes from your problem interpretation, your problem identification, and we need to learn to interpret that word. And what we've really kind of done so far is gone down this path of, photosynthesis, right? So we could define plant growth in terms of photosynthesis and really the rate at which photosynthesis happens, and we now know some of the environmental conditions that allow for that photosynthetic rate to increase or to maximize, or rather even optimize. But there are other things that you need to understand for growth too, and there are other concepts, right? So growth can be defined in multiple ways. One of the ways that it could be defined at the cellular level is that cells getting could be defined as cells getting larger in size, right? So cells could get larger, and really what you already know is that this can be determined with microscopic measurements, right? So think back to our previous project with the microscope design. When we were in lab, we were measuring our field of view, estimating cell size, and if you were to systematically, kind of like a catch and release even, although these plant cells aren't moving away, if you were to take plant leaves and put them under a microscope and measure the size of these cells, you might see over time that these cells get bigger. And conveniently, if you've got your microscope attachments ready to be printed, and if they function correctly, you'll actually be able to measure those cell sizes out in the lab, and that's a really great data point. So when you go to evaluate your prototypes, to test your prototypes, and to see if it is in fact growing, you could determine the rate of growth through some of the labs that you were given as methodologies for your data collection. You could also do some microscopic measurements, so you could look at the change in size of a cell over time, and that's going to give you your rate of growth. So that's one way you could do it, but there's yet another way to measure growth, and this is through cell division. And it's only in biology that we say that cell division is the same as cell multiplication, where one cell divides and we have two cells, right? So division and multiplication kind of mean the same thing in terms of biology, but clearly in math and algebra and arithmetic, that is not true, right? Division is not the same as multiplication. But really what we're going to do is really focus in today, in this video, on this process of cell division. And that's going to be key for us to understand what we're trying to talk about here. And I hope that you can think of a few key words that you might think about here. But I just want to give you some context to these, and then we're going to talk about this in terms of another diagram. So in cell division, there are three different types of cell division. And the first is binary fission. So binary fission, I want you to just Google that, look that up. Really, this is only in prokaryotic cells. And if you need a refresher on what prokaryotic cells are, you need to go back and look at your notes. If your notes are inadequate, you need to go back and watch a video and then update your notes. I expect you to know what prokaryotic cells are for your assessments. You've learned about them. You are expected to carry that knowledge forward, as well as your microscopic measurement information. So I expect you to be able to do those kinds of things on your upcoming cumulative benchmark on Thursday. The other type of cell division that you might be familiar with is mitosis, but I'm going to put a little asterisk next to that, because mitosis technically is not cell division, and we'll talk a little bit about what that is. And then you might have also heard of this, meiosis, which I'm going to put an asterisk as well, because it's also not cell division, but it's related to cell division, and oftentimes it's taught to you as cell division, but I'm going to convince you, I hope, that it's not quite the same thing. So before we talk about what mitosis and meiosis are, because what we're looking at are your plant cells, which determine your growth because we're growing vegetables, because those are plants are eukaryotic cells, we're really going to focus in on is mitosis in our discussion today. But mitosis happens in eukaryotic organisms, as does meiosis. All right. Again, review eukaryotic cells as well. So what I want you to do is draw a circle. I'm going to do the best that I can to draw a circle. Can't make any promises. Alright, so that's an okay circle. But now that you know how to do some geometric constructions, you could probably do a perfect circle. Right, so anyways, I'm just going to, this is for a note so I'm not so concerned about it. I got my center point there. But this is the cell cycle. Right, so what we're going to do is talk about the cell cycle, which is another way of saying the life cycle of a cell. All right, so the life cycle of a cell. So what we're going to do is divide this. Actually, I should do this as a solid line. And we're going to put some directional arrows. So we're going to say that this goes clockwise. And this part right here, let me do this in blue here. This part that goes in this direction right here is called interphase. Now, interphase is really when the cell does its main job, right? So interphase is key. This is when the cells do the job that they've been differentiated or signaled to do by reading its DNA. So leaf cells are going to be able to grow horizontally most of the time in order to maximize the surface area on them to get the most sunlight to do the highest rates of photosynthesis. In your body, your eye cells would be sensing light. During this interphase, your heart cells would be contracting their muscles so as to pump blood around your body. And so interphase is really a key thing because this is when the cell does its typical job. But this is actually divided into 3 subcomponents. So I'm going to do some dotted lines here because this is all 3 parts of interphase. But first comes G1, then S, then G2. And G1 stands for gap 1. This is G2 is gap 2, and S stands for synthesis. And that's going to be an important word that you're going to need to know over and over in terms of bio and chemistry particularly. Synthesis means to make something, right? If I'm asking you to synthesize something, I'm asking you to make something. And in this case, what we're talking about is DNA synthesis. So what's happening here is a process that maybe you've learned about, we will definitely learn more detail this year, is DNA replication, right? So making more DNA, making the extra strand of DNA. And a term that I want you to be familiar with, but we'll learn more detail about, is semi-conservative replication. And I want you to look up what that term means, semi-conservative replication of DNA. It's here in gap 1 that DNA gets read and codes for this RNA molecule that then codes for protein. And we're going to learn all about that process, this process right here, and this process in greater detail later in the year. But these proteins are critically important. So think back to the nitrogen cycle. These have nitrogen in them. They're made out of these things called amino acids. And they also, some examples of them are enzymes. So we talked about enzyme denaturation last class. These proteins do the job of the cell. They are the machines that keep things alive. And so when a cell starts here, that DNA, in order to be read, I'm going to do this in a different color, is in a form called chromatin. Okay? In chromatin. And I want you to ask me, or even better, ask Mr. Pankratz, my substitute, because we're going to be out on Tuesday. Mr. Pankratz, who is a biology teacher, was a biology teacher, what chromatin is. But that chromatin is, the DNA is in the chromatin form up until this point, right here. Right until this point right here, where it condenses and then goes into chromosome form. Okay? In chromosome form. So this next phase right here is also critically important. I'm going to divide it. I'm going to draw another solid line as best I can, and then we're going to do some dotted lines to break this into 4 sections. And we're going to label this green area mitosis. And that's where we have something called prophase, metaphase. I hope these are familiar to you. Anaphase and telophase. Telophase. And really all that mitosis is, I told you it's not cell division, it's nuclear division. Alright, so it's when the the nucleus divides into 2. And that's critically important because from that you should be able to infer that if it's about nucleus dividing, it's only for eukaryotic organisms, or eukaryotic cells, things with nuclei. So this is really when the nucleus divides, but the nucleus dividing is going to be critically important because the next thing that happens right here is cytokinesis. Cyto means cell, kinesis means movement. So in other words, the cells move apart from each other, the cell splits. So in mitosis, the nucleus divides, and then it's followed by cytokinesis, which is when the rest of the cell splits. So then you end up with 2 identical cells. Because DNA was copied first before it divided, you end up with 2 cells with the exact same genetic information. Then those 2 resulting daughter cells will repeat this process in their own cell cycle, their own life cycle, making copies of DNA, reading DNA, making copies of that DNA, and then splitting those 2 copies of DNA, and then the rest of the cell, and repeating. And that's really what I want you to make sure that you understand for next class. But we could actually measure the rate of growth, right? So if we're going back to this right here, if we're looking at growth here in mitosis, there is this idea, there's an idea here that's called the Mitotic Index. And all it is the number of cells in mitosis over total number of cells times 100. Essentially, that's all it is. So you're looking at the percentage of cells that are going through mitosis. If more and more cells are going through mitosis, and the reason we pick mitosis is because it's easy to see those chromosomes. Again, ask Mr. Pankratz what a chromosome is and how it's different than chromatin. But you can identify those cells, and if you can count those cells, you can see that there are more cells that are undergoing mitosis, which will then be followed by cytokinesis, and then, therefore, lead to more cell division and more growth. So cells getting larger is another way, again, that we can measure that, but there is a very important limitation of cells getting too large, and that those of you who came to the Excel workshop using the formula saw that as the surface area to volume ratio, well, as a cell or a shape gets larger, the surface area to volume ratio gets smaller, and we want a large surface area to volume ratio. And if you don't understand why or what that means, what that surface area to volume ratio indicates, I need you to ask me. Now, I'm also getting the sense that some of you are coming to ask me individually what these questions are, but I'd rather you ask me that in class where everyone would benefit from the answer. Some of you are also coming to me wanting to review your notes. Well, you should be writing what I'm writing and what I'm saying. And when I'm saying when I say to you, well, what questions do you have? I really am not asking you to tell me the questions I've told you to ask me. What I'm asking you to do is generate your own questions about things that you don't yet fully understand. I hope that's clear. I see that I'm at close to 16 minutes. I'm going to stop there. But you're going to be doing a little bit of a review in class on Tuesday, and you have a test on Thursday. And those things that are on the test, those content areas on the test, include nutrient cycles. They include all three of the nutrient cycles. You need to be able to draw them. You need to be able to explain them. You need to be able to interpret them. You need to be able to apply them. You also need to know water chemistry and the properties of water, photosynthesis, the overall reaction photosynthesis, the details of photosynthesis with the light-dependent, light-independent reactions, the graphs of how these these environmental factors affect the rate of photosynthesis, to explain those graphs, as we did in class, as well as the cell cycle. And you should be able to think about how this all applies to your greenhouse design and ways to measure and test and evaluate your designs. Okay? I think I'm going to stop there.